Welcome to class number 4, session number 3. This session will focus on sensors based on nanoparticles for the detection of DNA as well as biomolecules. One way to monitor changes in a solution of nanoparticles is by monitoring the changes in the optical properties of the solution. In the presented example, the changes in the absorption of the solution are examined. This system is designed in order to detect the presence of a specific DNA. For that purpose, the nanoparticles are modified through a thiol bond with a single-stranded DNA that is complementary to the targeted DNA. As seen in the right figure on the screen, the modification of the nanoparticles changes mainly the amount of the absorbed light by the solution, as compared to the unmodified or the bare nanoparticles, which have characteristic peak at about 560 nanometers. When adding the targeted DNA analyte in the solution, agglomeration occurs. Aggregation means that the nanoparticles come closer and closer together. This phenomena changes the absorption of the solution in several manners and can be seen in the graph. The location of the absorbance peak is an indication for the average size of the nanoparticles. When the peak is shifted to higher wavelength, namely from 560 nanometers to about 600 nanometer, it means that the average size of the nanoparticles in the solution is larger due to the aggregation. In addition, the width of the peak is much broader after aggregation, an observation that indicates for higher distribution in the nanoparticle size as compared to the reference solution or the nanoparticles. All in all, the presence of a targeted analyte, in this case the DNA, can be determined by monitoring the absorbance of the solution. With this in mind, monitoring the change in optical properties at specific wavelength, the sensing properties can be examined according to the following guidelines. The signal at 260 nanometers relates to the DNA molecules, and the signal at the 700 nanometer relates to agglomerations of gold DNA composites. As seen in the left figure, when heating the solution to around 80 centigrades, the target DNA dissolves in the solution, and its related signal at 260 nanometer increases. In a similar manner, the aggregates dissolve back to nanoparticles, and the aggregates peak at 700 nanometers decreases. This process is repeatable when heating and cooling the system. The change in the optical properties is also visible when looking at the colors changes of the solution. All in all, DNA-capped gold nanoparticles is a promising form for the detection of DNA strands throughout controlling the temperature of the solution of the DNA-modified nanoparticles. Using similar concept, a drop of nanoparticles can be casted on a paper. As seen in panel number A, when the sensor meets its target DNA, the color of the drop changes due to agglomerations. This system don't require sophisticated equipment for analysis and is appropriate mainly to determine the presence of an analyte and not its concentration. Detecting small concentrations of target DNA that don't create such equate changes is more challenging in these sensors. Panel B on the screen presents gold nanoparticles modified with short, single-stranded DNA to indicate the presence of a particular DNA sequence that is hybridized on a transparent substrate in a three-component sandwich assay format. At high target DNA concentrations, many nanoparticles are attached to the transparent surface. In this case, the high concentration of the nanoparticles cause a dramatic change in the color, and the substrate look pink even for the naked eye. At lower target concentrations, the attached nanoparticles cannot be visualized with the naked eye. To facilitate visualization of nanoparticle labels that are hybridized to the array surface, plating silver ions on the gold nanoparticles is used as a signal amplification. This way, very low surface coverage of nanoparticle can be visualized by a simple scanner or by just the naked eye. A similar principle is applied in the example presented in panel C, where the attachment of the nanoparticles to a substrate in a three-component sandwich assay format is detected by the change in the scattered light. A more applicable approach that the sensing method presented in panel C is monitoring the electrical changes of a sensor, while the sensing mechanism is similar to the previous examples in the previous slides. The setup in this sensing device is an insulating substrate with two electrodes for measuring the electrical resistance. On this substrate, nanoparticles are assembled in low concentrations, so there is no direct connection between the nanoparticles, and therefore the electrical resistance is very high. If DNA is captured, silver will plate on the nanoparticles. This plating will contact between the nanoparticles, and the electrical resistance will dramatically decrease step by step. By measuring the changes in the electrical resistance, a sensing signal can be obtained and monitored. Different approaches that utilize similar sensing mechanisms but use different characterization methods or instrumentations has been developed and tested to detect the attachment of nanoparticles to a substrate. One way is to monitor the surface-enhanced Raman spectroscopy signal that can be read throughout Raman spectroscopy, as depicted in panel E in the presented figure. Another way is to monitor the diffraction of a laser beam when the laser beam is directed towards the nanoparticles that are attached to the surface, as also could be demonstrated in the schematics on the slide. A very sophisticated sensing approach that allows sensing tiny amounts of targeted molecules inside the human body or in complemented environments relies on magnetic nanoparticles. One example of such sensing systems is based on magnetic microparticle probes with antibodies that specifically bind a target of interest, and nanoparticle probes that are encoded with DNA that is unique to the protein target of interest to be detected. In this configuration, magnetic separation of the complex probes and target is followed by dehybridization of the DNA on the nanoparticle probe surface, thus allowing one to determine the presence of the target proteins by identifying the DNA sequence released from the nanoparticle probe. To demonstrate the sensing process that relies on magnetic nanoparticles, I present here a short video that is taken from the following link. The general concept of this video is that separation of molecules and afterwards sensing can be done by magnetic nanoparticles. The solution contains many molecules, and this system is a sorting system that can be select and separate target molecule. The separation process includes 3 main steps. Step number 1 is that magnetic nanoparticles are connected to organic molecules that can absorb specific target biomolecules, and lock and key principle are added to the solution. The step that follows this initial consideration is that magnetic force is collecting all the nanoparticles to a specific location, and in this case the solution with the contaminating molecules can be removed. Right now we see on the screen step number 2, where the magnetic force is collecting the nanoparticles to a specific location. Afterwards, a new pure solvent is added and the wanted molecules will diffuse or dissolve in the solvent. The magnetic collection of the nanoparticles is repeated, but this time the particles are not connected to the target molecules, and those can be isolated. This is the end of class number 4, session number 3. Thank you.